Wednesday, we ended class right before getting to the specifics of translation. All right, so we're going to start by talking about translation today. All right, so just to, again, reorient yourself here. We are talking about the central dogma. We are talking about the portion where RNA gets translated into a protein and essentially are expressing those genes that are being translated. All right, so there are 3 stages of translation, and you will be happy to know that they are called the exact same thing for transcription and translation. All right, so it is initiation, elongation, and termination. Right, same 3 stages that transcription has. Right, if we look at our image here, right, I think it's kind of a fun little image here. This kind of reminds me, I don't know if you've seen images of trees eating things. All right, but that's what this image reminds me of. The ribosome over top of our mRNA is like this tree eating this fence post. All right, so if we talk about the 3 stages, all right, initiation. Right, this is when we actually start. My pointer's not working yet. I don't know why my pointer is so temperamental, but it's driving me crazy lately. So we start with the first step, in initiation. Right, mRNA is going to bind to the small ribosomal subunit. So remember, ribosomes have 2 subunits, large and the small. It's going to bind to this small subunit, right, via hydrogen bonds at this ribosomal binding site. Right, so small subunit is binding to our mRNA. Right. The codon, the start codon, which is AUG, is just a little ways downstream from that binding site. Right, so it's perfectly positioned to start. The next thing that's going to happen is this initiator tRNA is going to come in. Right, this initiator tRNA carries a modified amino acid that basically signifies that this is the start of our polypeptide. Right? We can see that initiator tRNA binding to our mRNA. And then finally, that large ribosomal subunit is going to come in and bind to the small subunit. So we have our full ribosome ready to start with our translation. Right? So at the end of all of this, right, our ribosomal subunits are put together, and our initiation tRNA, or initiator tRNA, is in this P spot. Remember, we have those 3 locations on our RNA, the E site, the P site, and the A site. Right, to start with, our tRNA is in this P site. There is a slight difference here between how bacteria and eukaryotes carry this process out. Right? The only slight difference is that in eukaryotes, instead of having this ribosomal binding site in the RNA sequence, it's simply going to bind to that 5' cap that we talked about, those modifications that happen after transcription. Right, so adding on that 5' cap, that is where this ribosome is going to bind instead. All right, so everything is bound to the mRNA. We are ready to go and start with our translation. And so our next step, elongation, this is where we're actively growing that polypeptide. Right? We're adding new amino acids. And so what's going to happen is we're going to get a new tRNA that is charged. Right? Remember, meaning that has an amino acid attached to it. That's going to come into this A site. Right? Now, this anticodon, right, is complementary to the codon in the mRNA, which signifies which specific tRNA is going to come in. Right? So that tells it which specific amino acid connected to this tRNA needs to bind here. A polypeptide bond is going to then be formed between the amino acids at the A site. Right? So if we look at this image here, we have our growing polypeptide that was at this P site, right? But this next step, we're taking that polypeptide and we're moving it over to the A site and binding it to that new amino acid that had just come in, right? So number 3 on my bullet points is basically saying that same thing again, right? The polypeptide is removed from the tRNA in the P site and it's transferred to that new amino acid in the A site. Right? This specific reaction is called peptidyl transfer reaction. Okay. So this is going to continue going on until all of the mRNA is read, right? Once we have bound a new amino acid, this ribosome is going to shift over by one codon, right? That means that the tRNA that gave away that polypeptide chain is going to shift into this E spot, and it's going to leave the ribosome, right? This tRNA will then go on to get another amino acid bonded to it and go through this process again. A new charged tRNA, right, one that has an amino acid, is going to come into our A spot, and this whole process is just going to keep on running. Now, when we get to a stop codon, right, this is when we have this factor called a release factor coming into play. So instead of a tRNA, right, this stop codon instead codes for this release factor, which binds to the codon at the A site, right, and tells transcription to stop. Right? So once it's bound, right, the bond between the polypeptide and the tRNA is going to be broken. The RNA is going to be freed. The ribosome is going to break apart into those subunits, and our factor here is going to be released. Right? And what results is this chain of amino acids. Right? Now that amino acid chain is going to go on to form its 3D structure. Right? It's going to form all those different three-dimensional structures, the tertiary, the quaternary structure, to become a functional protein after this. Right? But at the end of this stage, right, we have made a polypeptide. Polypeptide that will then go on to be folded and become active. All right. I think we need a visual to solidify this, right, and see it all in action. So we have this nice video here. Oh, this video does not have sound. I forgot about that. So our tRNA is coming into the P site. At the A site, the amino acid is being transferred to that new amino acid, and that polypeptide chain is growing. The ribosome is shifting down one codon at a time. Right. When we get to the end here, we have that polypeptide chain. This release factor comes in, the chain is released, right? And then everything can basically break apart back into their subunits, and that is the end of translation. Right? So I think this is a really nice visual that helps you visualizing how everything's moving and shifting and happening kind of all at once here. All right, but if we zoom back out to big picture, right? We have talked about transcription. We have talked about translation. So we have talked about DNA becoming RNA and eventually becoming a protein. Right? Proteins are important because when that trait is expressed, we get the phenotype. Right? And I really like this image here because it shows a phenotype that's easy to see with the naked eye. Right? So this would be fur color. And we can see that these different fur colors, they're transcribing a different protein, which leads to a darker mouse and a lighter mouse. Right? So keep in mind that there's a bigger picture to all of this. Right? The underlying DNA code is influencing the traits that are actually showing up in our organisms. All right, so let's do some class review before we move on to the next topic here. So I have a few questions here for you to answer. One is thinking about amino acids and how many nucleotides might be present to code for those amino acids. And then talking about the different stages of translation. So I'll give you a couple minutes. I will finish passing out some papers too. Is not too far away. I promise. I myself am looking forward to Thanksgiving break. Alright, if we think of these questions here, the first question is talking about insulin, right? In both humans and horses, right? Insulin both have two polypeptide chains. One chain has 21 amino acids, and the other contains 30 amino acids. How many nitrogenous bases are present in the DNA code for each chain? What do you have to do to figure this out? I see it being mouthed. Yes, multiply it by 3, right? Each amino acid has a codon, and a codon is 3 of those bases. And there we go. All right, so you'd have to multiply it by 3, right? Each amino acid is coded for by a codon, which is 3 nitrogenous bases. So for 21 amino acids, we would need 63 bases. For 30 amino acids, we would need 90 bases. What stage, right, of our 3 stages of translation are both subunits of the ribosome assembled? By show of hands, is it the first, the second, or the third stage? Yes, it is the first stage, right, which is initiation, right? That small subunit is going to bind. That tRNA is going to come in and bind to the mRNA, which then signals for the large subunits to bind to the small subunit. And then which site in our ribosome, right, the E site, the P site, or the A site, is the polypeptide chain actively growing? We have votes for P site. Do we have votes for any other site? It would be the A site, right? It gets transferred to the P site, but as it actively grows, right, that polypeptide is going on to that A site, right? It's being bound to that new amino acid that comes in at the A site. Any questions about translation? All right, then we're going to shift to an entirely new topic. I guess not entirely new. It's still in the same vein of thought here. But we're going to talk about mutations and DNA repair, which I really like talking about mutations. I think they're really interesting. All right, if we're talking about mutations, right, specifically mutations in DNA, we're talking about a random change in the nucleotide sequence, right? Keyword here being random. Right? Organisms are not choosing to have a mutation. Right? They are not specifically influencing a mutation occurring, but this is completely random. Right? So if we look at these mutations, they can have 3 potential effects. They can either be beneficial, they can be neutral, or they can be deleterious, so they have harmful effects. If it's neutral, a good example would be like this flower up here would be like a neutral effect, right? One of the mutations is causing one petal to be yellow instead of red. Most of the flower is red. It's likely not going to have a harmful effect on pollination or reproduction. If it's harmful, a good example is in this fruit fly here, right? It has a mutation in the gene for the antenna, which makes it have this leg instead of an antenna in that position. You can imagine how that would be harmful for the fruit fly. A beneficial mutation, one we've already talked about way in the beginning of class, lab number 1, when we talked about sickle cell disease. Right? We talked about a mutation that caused the change of an amino acid, right? And that change of amino acid can cause sickle cell disease, right? Which is not always a bad thing, right? In fact, it can be beneficial if you carry this disease but don't express the disease, because it makes you have a better defense against malaria, right? So this is a good example of a beneficial one. There are many other examples out there. Okay. Let's look at some of these very specific examples of mutations that can occur. And we're going to look at point mutations. And the point mutation is going to be an alteration in the DNA code at a singular base pair. So we'll see later on here how a singular base pair can become a few base pairs at a time, right? But broadly speaking, point mutation is a mutation in the DNA code at a single base pair, of which there are 3 types, all right? We can substitute a base pair, we can insert a base pair, or we can delete a base pair. We're going to start by talking with the substitution first. Okay? So in a substitution mutation here, one base pair is going to be swapped out for another one. Okay? A really good example of this is a process known as spontaneous deamination. Okay? It's this interesting phenomenon that occurs where spontaneously, okay, we can have base pairs changing, say, from a cytosine to a thymine. Right? This happens spontaneous. Right? There's no precursor. There's not any mechanism in the DNA that tells it to do this. All right? It's fairly common, right? And most of the time, this will be picked up on during DNA synthesis and repaired. Right? But sometimes it's not repaired, and we see this substitution and mutation occurring. All right, so all of our substitutions, we can break them down into different types. And so if we look at our DNA code here, our sample DNA code, in yellow here we have our mRNA. We would call that the wild type allele, or the normal allele, right? What we normally would find. And then the proteins that it encodes for is in purple below. Right. One of the first mutations that could occur would be a silent mutation, right, or a synonymous mutation. So in this type of mutation, right, it's still a substitution mutation. We're still swapping out a base pair for a different base pair, right, but that swapping has no effect on the amino acid that it codes for. Right. So if you remember looking at that codon chart, right, the codon chart is redundant, which means there can be multiple codons that code for the same amino acid. So in a mutation that is silent, right, we have this mutation, but it still codes for that same amino acid, right? So there's no change. We can also have a missense mutation, okay? And this is where we are swapping out a nitrogenous base here for a different one, right? But this time it leads to a different amino acid. Right, so if we swapped out our base here and made it an A, it's going to code for an entirely new amino acid. Right, slightly in the same vein here, we have a nonsense mutation. Right, so still swapping out one of our nucleotides, right? But this time that swapping, instead of causing a different amino acid to be coded for, it's going to code for a stop codon. Right? So the result would be that our translation would prematurely stop at the stop codon instead of translating the rest of our code. Right? Likewise, we can have a sense mutation, and I don't have an image to correlate with this one here, but this one's going to change a stop codon to a non-terminal codon, to an amino acid. So instead of stopping where it should, it's going to continue on. Right? So these are 4 different types of our substitution mutations here. Right? Again, we're just swapping out one base pair. We're not making mRNA longer or shorter. We're just substituting what nucleotide base is present, which can either code for a new amino acid, a stop codon, prematurely stopping or not stopping at all, or it could have no effect whatsoever. And this is where we get the importance of these wobble positions. So these codon wobble positions. So that would be the 3rd nucleotide in a codon. Right? So if we look at our codon chart, we see many, many, many instances of multiple codons coding for the same protein. Right? So this third position is often the one that is different than the other two. So for example, for serine here, they both start with UC, right? But that third position could be different. Right? And this is important because often where a mutation will occur is in this third position, right? When a tRNA comes in, right, the base pairing is looser between the tRNA and the mRNA in the third position, and it's more often likely to make a mistake, right? And so this is where this wobble position is very beneficial, right? This redundancy in our amino acid code. Plus, I think it's fun. We talk about wobble positions, right? It's kind of a fun word to say. All right, so we talked about substitution mutations, but there are two other types of mutations here, insertion or deletion, right? And this is where the point mutation definition will be expanded upon a little bit. So a point mutation is an alteration of the DNA code at a singular base pair, right? While an insertion or a deletion, we can insert or delete more than one base pair, all right? So we're either adding or subtracting in our DNA sequence here, right? This is often going to result in what's called a frame shift mutation, right? So a frame shift mutation is going to change how we read our mRNA and the amino acids that it's coded for, right? So if the addition or deletion of a number of nucleotides that are not in multiples of 3, right? So it's not in a multiples of 3. If it was in a multiples of 3, we'd see that it either add or delete a single amino acid, right? But if it's only 1, 2, or 4, 5, that's going to shift how we read our frame, right? So if we pretend that this sentence, each of those letters are amino acids, right? We could read it as the fat cap set, right? But if we were to delete one of those amino acids, and we're deleting that first one, that T there, it's going to shift how we would read it in the sense of codons. Remember, codons are always read in pairs of 3. So if we delete out that T, all of a sudden we have a sentence that makes no sense at all, right? We are shifting how we read it. So if we look at an actual DNA sequence, right, our normal DNA sequence here has our 3 codons and encodes for our 3 amino acids. If we were to insert an additional amino acid in here, right, remember we're inserting it, we're not substituting it. So it's one extra nucleotide here. It's going to shift how this is being read, right? So the first pair is read normally, right, coding for the same amino acid, but our T is being inserted here, which shifts how it's read. So now we're coding for different amino acids, and in fact, we actually have this extra T at the end, which doesn't code for an amino acid because it can't, right? Because it's not in a set of 3. It's not a full codon. So we're shifting how we read it. Right. Now, if, for example, we were to insert 3 bases, right, or 3 nucleotides into a sequence, this would not be a frame shift mutation, right, because it's going to encode for the same amino acids. We're either going to add in an amino acid, like in this case, right, or we're going to take out an amino acid, right? But all the other amino acids will be read normally. We'll just have an insertion of some random amino acid because of this triple insertion here. Right. The same slide, we're going to walk through it again just with some different examples. Right. So again, we have our wild type or our normal allele here and the proteins that it normally encodes for. Right. If we insert or delete some base pairs, right, not in multiples of 3, we're shifting the way that it's read. So this first example, we're taking away a uracil, which shifts how it's read. So the first 2 are the same, but once we get to the point where it was deleted, we see 2 new amino acids in this place. All right. Same if we were to insert maybe this U somewhere else, Right? In this case, you see we're inserting a U. The first amino acid is the same, but now it's actually coding for a stop codon. Right? So it's going to prematurely stop reading the mRNA because of the frame shift. Right? Again, likewise, if we add in 3, right, multiples of 3, it's not going to shift how we read it, but it's going to add in an additional or subtract an additional amino acid here. So in our example here, we are removing 3 bases and we're subtracting one of the amino acids that should be there. All right. Any questions? All right. We're going to work through this problem set here. That should probably take us up to our content check. So in this table, I am giving you the original strand, the original DNA here, right? And in the resulting 4 lines, right, a mutation has occurred. I want you to tell me the name of the mutation, right? Define what that mutation does, and the consequence of that mutation. Right? I'll give you a few minutes to do so here. Now, I'm actually going to work through this too because I did not put the answers in my slides, and so I need to figure out the answers as well. Now remember, you are going to have to convert this to mRNA. This is a DNA strand. It's being converted to mRNA. So remember, T is substituting, or U is going to substitute for where a T would be. Did you just whisper my name? Oh, I thought I heard my name. I put 2 minutes up here. You might need more than 2 minutes, and that's okay. Yes. I'm going to say maybe take another minute to work on this, and then we will start off with this slide again on Monday, right? Because my alarm's going to go off in a second. Skip to the content. It definitely takes more than 3 minutes to solve. All right. I think we're going to switch the content check now. I turned off my alarm, so I'm going to startle anybody here. But this is a really great example of a question that I could ask on an exam, right? I'm giving you the DNA strand. You have to change it into RNA, right? So basically you have to transcribe it, and then you have to translate it several different times, and then figure out what the mutation was and what it did. Right? If I were to ever give you a question like this on the exam, right, I would give you the codons and what they stand for, what amino acids they would code for. I would not expect you to memorize that codon chart. All right, let's skip ahead, and we'll go over the answers to this on Monday because I know some of you are still working on it. It'll give you a chance to do some good review over the weekend if you want to study for bio. Let's go to the content check. Actually, before I do that, study guide is up on Moodle, right? So study guide is available. All right. So the content check is kind of similar to what we were just doing, all right? I want you to use the DNA sequence that I'm giving you on the bottom here. I want you to transcribe the corresponding mRNA sequence and then translate it to the proper amino acid sequence, right? These do not have any mutations in it. I simply want you to transcribe it and translate it. So I'll give you a hint to start with. Right? Remember, RNA polymerase can only read in a certain direction, so it's only going to transcribe one of the strands. Yeah. I handed it back to him like,